Hello, everyone. My name is Priya Prakash, and I am a graduate student at the Interdisciplinary Life Sciences program and at the Department of Chemistry at Purdue University. My research is focused on modeling and characterizing microglial cell states in health and disease using various chemical perturbation methods. I am advised by Dr. Gaurav Chopra at the Institute of Drug Discovery at Purdue University. First and foremost, I would like to thank the Alzheimer's Drug Discovery Foundation for this wonderful opportunity to attend the 19th International Alzheimer's Drug Discovery Conference in New Jersey this year. It is truly an honor and a privilege to present our work and receive constructive feedback from colleagues and researchers at the meeting. It is also a great platform for young investigators such as myself to network and initiate collaborations with a common goal of finding new and innovative ways to advance Alzheimer's research. With that in mind, I would like to highlight that today I will be talking about developing new tools to study microglial exhaustion and microglial phagocytic capacity. So first and foremost, a little background on these mysterious yet magical immune cells of our brain called microglia. These cells originate in the yolk sac during embryonic development, during which time they migrate and reside in the brain during early embryogenesis. These cells are a very heterogeneous population of specialized macrophages that perform several functions to maintain tissue homeostasis during health and different diseases. Some of their functions are beautifully illustrated here. Microglia are involved in maintaining neuronal plasticity. They also play a big role in synaptic pruning, as demonstrated by Dr. Beth Stevens and several others in the field. These cells, when activated, also release inflammatory cytokines that can go on to activate other cells in their environment, such as astrocytes and oligodendrocytes. And finally, their function that we are mostly interested in is their phagocytic function. So phagocytosis is when microglia recognize and engulf either dead cell debris or toxic peptides from their microenvironment. We all know that during Alzheimer's disease, amyloid beta peptides start accumulating in the brain, and that causes cognitive decline and neuronal death. During the early developmental stages of the disease, activated microglia readily engulf and degrade the amyloid beta particles. However, it has been shown that in aged brains, over time, microglia lose their phagocytic capacity and they become functionally impaired in Alzheimer's disease. With this in mind, our central hypothesis is that a subset of these microglial cells become exhausted due to their chronic exposure to amyloid beta, which results in their loss of phagocytic function. Now, in order to study this, we require specific tools, A-beta related specific tools, to model this in vitro and in vivo. The current limitations of the available tools to study phagocytosis is that fluorescent latex beads or pH-RODO-labeled E. coli and zymosan particles do not provide the amyloid beta target specificity to these cells. Furthermore, the fluorescent-labeled amyloid beta exhibit background fluorescence, and it is also difficult to differentiate between amyloid beta that has been internalized by the cells versus amyloid beta that is stuck on the cell membrane. And due to this, it is difficult to sort and analyze these live cells for downstream applications. To address these limitations, we have developed a pH-dependent fluorogenic and magnetic amyloid beta peptide conjugate that exhibits maximum fluorescence at the acidic pH and limited fluorescence in the physiological pH of 7.4 outside the cells. The assay principle is described here. When the peptide is added on live cells in culture, the phagocytic microglia that uptake these cells exhibit green fluorescence due to the presence of the acidic phagolysosome within the cell, and the cells that do not uptake these peptides do not exhibit fluorescence. So this now is a great way to sort these live phagocytic and non-phagocytic cell populations for downstream analysis and applications. This has been validated at different concentrations that I will show you in a while. And the magnetic bead labeled amyloid beta can be sorted in a similar way, but using a magnet. When you apply a magnet to the mixed cell population, the phagocytic microglia that have uptaken the magnetic labeled amyloid beta get sorted with the magnet as the pellet, and the non-phagocytic microglia that do not take up the A beta remain in the supernatant. So now the different cell populations can be sorted, and applications like single-cell RNA sequencing can be performed to study these cell states at different conditions. I would also like to highlight here that for drug discovery, these sensors can be used to use different small molecules or drugs to demonstrate or to measure enhanced phagocytosis that is required for the assay. To validate these pH-dependent amyloid beta sensors, I have characterized these sensors in live microglial cells in both the mouse or the murine microglial cell line, that is BV2 and N9, and the human microglial HMC3 cell line as shown here over the course of a 24-hour live cell real-time assay. As you can see by the red arrows, cells that take up these pH conjugated amyloid beta peptides exhibit green fluorescence, and the non-phagocytic cells, some of the cells in these photos do not exhibit any green fluorescence. We can go on to quantify this in live cells by counting the number of cells that exhibit fluorescence versus number of cells that do not at different concentrations with the addition of different small molecules. And what we observed just as a proof of concept in culture is that microglia exposed to lower and higher doses of amyloid beta sensors exhibit varying phagocytic capacities over a 24-hour time period, possibly due to exhaustion resulting in loss of function. Current work in this area is in using primary microglial cells from both mouse and rat brains, and measuring their phagocytic capacities ex vivo. Furthermore, our pH conjugated amyloid beta retains their fluorescence in fixed cells. So this is a great way to use antibodies to identify protein expression in cells also. And we have confirmed the intracellular localization of our amyloid beta in the acidic phagosomal compartments within the cell by using the red lyso tracker here on the right. And our amyloid beta exhibits green fluorescence, and the merged images show the co-localization of our amyloid beta with the lysosomal or the phagolysosomal organelles within the cells. So the ongoing work, however, is now that we have these tools with us, amyloid beta specific tools, the questions that we ask are related to the microglial cell states in different disease and in health conditions. First of all, we can ask the main key question, that is, what are the genomic or the transcriptomic differences between the amyloid beta specific phagocytic versus non-phagocytic cell populations? This will give rise to new targets for Alzheimer's disease, and new targets will lead to new small molecules and new drugs that can be used to target these proteins. So I have shown as a proof of concept that in primary microglia in adult from adult mouse brains, our pH labeled amyloid beta works really well, as shown in the image on the right. Current work is in sorting these live cells and asking questions ex vivo regarding their ability to produce different cytokines under different small molecule conditions. And of course, the main future goal will be to use our sensors in vivo in mouse and rat models to really sort these different cell populations and do a downstream assay like a single cell RNA sequencing to identify new targets for Alzheimer's disease. With that in mind, I would like to thank my advisor, Dr. Gaurav Chopra, and my mentor, Dr. Krupal Jethwa in the Chopra lab, along with our collaborators, and of course, the Alzheimer's Drug Discovery Foundation for giving me this platform to talk to you all about my work. Thank you.